All right, so since we are now moving to an online format, we will post this video in lieu of meeting for lab class in person. So we're going to discuss PCR and gel electrophoresis. This is antibiotic resistance week 3 and 4. So let me just remind you of the flow. We're in the middle of this 4-week antibiotic resistance module, and since we had Tuesday class, you all, in this video, I'm going to cover week 3, which we would have done on March 24th. This is where we would have set up our PCR reaction. Remember, you all did streak plates to isolate colonies. I would use those colonies, or we would use those colonies and set up PCR reactions. We would then read the minimum inhibitory concentration, the MIC plates that you all set up. Remember those large antibiotic strips you all placed onto the bacterial lawns that you made? And then we would continue working on our PowerPoint. Then in week 4, we would have come in to analyze the PCR reaction. We have to run an agarose gel to visualize the PCR reaction results. And then we would finish data analysis, finish our oral communication, and then present on all 4 weeks of the antibiotics resistance module. So you all will still do this. I'm just going to give you a video to sort of describe what's going on. We are going to post the data. We've got pictures of all of these MIC plates posted for you for every group. You'll find those on Canvas. We ran the agarose gel for you from the PCR reactions that we set up. And we posted a picture of that also. That will be on Canvas as well. Okay. More information will be coming about the final presentation, so that won't be until April 14th. In short, I'll go ahead and just tell you, you will still do your final presentation with your partner. It just won't be done in person. Instead, you will put together a PowerPoint presentation and turn that in on Canvas. You'll still do a 1-page summary. And you'll peer review your partner's contribution, but all of that will be turned in on Canvas. All right, so learning objectives, of course. This one's all about the PCR and gel electrophoresis. We've been working with antibiotic sensitivity and resistance using the Kirby-Bauer, and the MIC experiments. Now we're going to take it a step further and look at PCR to do some genetic characterization. If we were in class, we would have learned how to use micropipettes because those are required. Those pipetters are required to set up the PCR and gel reactions. These are some more detailed learning objectives for you all as far as this module goes. We do want to add in some more details about the genetic makeup of bacteria because this is a genetic characterization of the bacteria. That's what PCR is doing. So we want you to understand the difference in the genetic makeup of the transformed strain. Basically, it has the transferase gene compared to the wild-type E. coli, which does not have the transferase gene. We want you to understand how PCR works at the molecular level. Remember, you did watch a video and answer a question set about this. Also, gel electrophoresis, similar. This was also in the video and the question set. Also, along the way, you all will be calculating the minimum inhibitory concentration of aminoglycosides and non-aminoglycoside antibiotics for both the wild-type and transformed E. coli strains. And then learn, of course, how we can use PCR and agarose gel electrophoresis to characterize the genetic makeup of strains. And learn how to look at agarose gel pictures and interpret those results. All right, so before you all left for spring break, you did set up the MIC plates. So you can go ahead at any point. You can get with your group online through GroupMe or through some sort of chats or text or email or whatever, or even using the online Google Sheets and the PowerPoint, docs or whatever. You guys will collect the data. You will analyze the data for your assigned antibiotics. Remember, you had one aminoglycoside and one non-aminoglycoside. You will discuss and analyze this data and add it to your growing oral communication PowerPoint. So what does the MIC data look like? I don't have it all up right now for you, but there will be something similar to this on your Canvas site. that's just probably been added. It'll say online lab resources, and there will be, this is of course Alexandra 1220 class. This may not be your class, but this is one of the classes, and this will be what yours looks like. It'll be specific for your class. Then there will be a PowerPoint file with every group, there. I want to show you some of them just to give you some ideas on what it looks like. Let me open up a couple of them. Hopefully it won't take too long on me here. Come on. This way I can show you some of these MIC pictures. Yeah, these take just a minute just because there is quite a bit of data in there. Here we go. We're about to get one. I apologize for this. I should have had one already up. Come on. All right. So here's an example of what you will see for the MIC. You'll see your little zone of inhibition. What you're going to be doing, you've got the ruler there to give you an idea if you wanted to sort of see the scale. But what you're going to want to do is read the concentration there where the zone of inhibition actually intersects with the strip. In this example, it looks to me like it intercepts right around between 8 and 12, so I would read that maybe about 10, and I believe that's micrograms per milliliter. And so you will just read, there's going to be replicates. Remember, you all did 3 replicates of each of your strips, so there's, in this case, 3 replicates of the gentamicin. Gentamicin is the only one, if you were assigned gentamicin, it's a little bit weird. The others are all very clearly labeled. Gentamicin says CN at the top of the strip. So these are all 3 gentamicin replicates. Cipro says CIP. You notice it's got a much bigger zone. CIP, CIP, CIP. Here is a control. So at the end there's a control picture. And then at the very end of that is a picture of your streak plate so that you can see what your streak plate looked like and you can see if you got isolated colonies or not. Another example of a PowerPoint, and one of the reasons I wanted to show you this, is that, so this, yeah, it's for this one. Some of these antibiotics worked really well against the bacteria. If you had a situation like this where the actual zone of inhibition or zone of clearing goes to the bottom of the strip, we're going to read this as 0. We're going to read this as 0, all right? That's what you would enter into your spreadsheet. Now, in addition, and I didn't open up one that shows this, but maybe let's imagine you do see one like this for your antibiotic strip, because some of them don't have a zone of clearing. There's resistance. In this case, we don't know what the upper limit is because it doesn't go high enough. So what we're going to do in this case would be to read and record the number at the top of the strip, the highest concentration number. So if there's no zone of inhibition, you will read the highest concentration number. All right, so go to your Canvas site, open the MIC plate pictures for your group, record and analyze the data for your assigned antibiotics, and add that to your growing PowerPoint. All right, and then let's start thinking now, what will we do to characterize the transformed and wild type strains genetically? So we're going to do some genetic analysis. This is where PCR is going to come into the picture. PCR is like DNA replication in a test tube, but it is not just random DNA replication. This is controlled DNA replication. We can control the region of DNA that gets replicated. And we do that by supplying the primers for the reaction. The primers will have a specific DNA sequence, and they will anneal to the gene that we are interested in. And so the primers we're going to use will be for the transferase gene. All right, and so if the transferase gene is present, the primers will anneal, and we'll get DNA replication of the transferase gene. if the transferase gene is absent, as it is in the wild type strain, there will be no way that the primers can anneal. So the primers can't anneal. The gene is not there. So the primers won't anneal, and we won't get replication. All right, so what is DNA replication? DNA replication is when a template strand of DNA separates and enzymes come along, and actually, in this case, we're going to use, there's going to be DNA helicase. Helicase will actually open up the template strand. It will break the hydrogen bonds and open up the template strand, creating this Y, or what we would call a DNA replication fork. You're going to see an enzyme called primase. Primase will come in and lay down an RNA primer. The RNA primer is really important, and because DNA polymerase cannot replicate the DNA without the RNA primer. And then DNA polymerase comes in after after primase has laid down the primer, and DNA polymerase then builds the homologous strand. Remember, you watched a video of this prior to coming, or prior to this module. Now, PCR basically is going to be DNA replication in the tube. All right, we have to add everything into the tube. We have to add our DNA template. We have to add in our primers. We have to add in our DNA polymerase. We have to add in nucleotides, the A's and T's and C's and G's. All right, so we add everything into the tube, and then we put it in a machine. I'll show you. All right, like I said, PCR won't just replicate any old DNA. We will use primers that anneal to the transferase gene only. So, in this way, PCR will tell us what strain we have. We will have amplification if the transferase gene is present. So PCR reaction will work in the transformed strain. But wild type does not have the transferase gene, and so no replication will occur. So the PCR will not work. All right, electrophoresis will be done afterwards so that we can visualize, because we don't know just by looking at our test tube if the DNA was replicated or not. All right, we have to actually then run an agarose gel to visualize, to see, is there DNA in our tube or is there not? All right, so let's look at a visual of PCR. And this will give us a really nice kind of understanding of all these pieces that might seem so confusing at this point. All right, so remember, PCR is DNA replication in a test tube, and it is done in what's called a thermocycler machine. The thermocycler will basically just cycle between these temperatures. It's going to go to a denaturation phase where it gets really, really hot, like about 95 degrees is what we're using. This will melt the hydrogen bonds. So this cycle basically takes, if we're thinking about DNA replication, this is what helicase would do. DNA helicase opens the template, breaking the hydrogen bonds. Denaturation does that. All right, then the machine will quickly cool down to an annealing temperature. Ours is going to be about 56 degrees. This will allow our specific primers that we supplied. These primers that we are going to put in are going to be complementary to the transferase gene. And so at the annealing temperature, the primers will anneal if the gene is present, because remember, it has to be, it's complementary to the transferase gene. If there's no transferase gene here in our template, then these primers can't anneal. But if the transferase gene is present, they will anneal when we go to the annealing temperature. And then when we go to elongation, what will happen is DNA polymerase that we add into the tube will take the nucleotides that are homologous, and it will basically bind to the end of this primer, and it will extend the primer. It will continue replicating the transferase gene, and you will now have 2 copies of the transferase gene. Then this thing will repeat and do it all over again. All right, it will just go over and over and over. Here's a schematic to sort of show you. You start out with 1 copy of your template. When you get denaturation, annealing, and elongation, it results in 2 copies. Then it repeats again, and you get denaturation, annealing, and elongation for those copies, and it turns those 2 now into 4. Then it repeats again, and you get now 4 templates that are copied into 8, and the cycles repeat again, and 8 becomes 16. After you do this many, many cycles, about 30 or so cycles, you will end up with millions of copies of the transferase gene if the transferase gene was present. Now, this will work, and this reaction should be happening in the transformed strain, but it won't work and won't be happening in the wild type strain because there will be no transferase gene for our primers to anneal to. All right, so here's how we're doing this. We picked colonies from Ural's plates that you made. We put them into the PCR tubes with the primers, with the DNA polymerase, with Taq polymerase, with the DNTPs, the A's and C's and T's and G's, and then we put lids on them and we put them into the thermocycler. The thermocycler then began going through all of those cycles over and over and over, and we amplified the transferase gene. Excuse me, this is not phosphotransferase. Ours was an acetyltransferase, wasn't it? So we will amplify the transferase gene in the transformed strain, but we will not amplify anything in the wild type strain. Then we will take our samples from the tubes and put them onto an agarose gel in order to visualize our results. All right, remember, the wild type strain does not have this plasmid, but the transferase strain, the transformed strain does have this plasmid. The transformed strain has, in this area where it says inserted gene, that's where the transformed gene is. You can see there is a primer site and a primer site on each. There's one primer site here and one primer site there on each side of our inserted gene. Excuse me, goodness gracious. Go back. And so when we allowed annealing, that's where our primers annealed, and then when we had extension or elongation, this is the area of DNA that was amplified. So our transferase gene is amplified in the transformed strain. All right, you need to think about this. Think about the genetic makeup, knowing that the transformed strain has a plasmid with the transferase gene there in this blue region, and knowing that the wild type lacks this and does not have this plasmid at all. Discuss with your group members electronically and come up with a hypothesis and prediction for the PCR reaction experiment. Think about it. What will you see on the gel? Will there be DNA amplified in the wild type strain? Will you see anything on the gel? Will there be DNA amplified in the transformed strain? What will you see on the gel? All right, if you had come to class, we would have been using micropipettes to set up these reactions. Our PCR reaction specifically had quite a few steps. We had to pick a colony from the agar plate using the pipette tip. We transferred the bacteria to a 1.5 mil Eppendorf tube containing 50 microliters of sterile water. We pipetted up and down to resuspend the cells. We made 2 tubes, one from each plate, so we made a wild type strain tube and a transformed strain tube. We lysed the cells to rupture them and release their DNA for 5 minutes on a heat block. We centrifuged then to remove cell debris, and we transferred 25 microliters of the supernatant, which hopefully either contained our DNA or did not, depending on which strain it was, and we moved it into a PCR tube. Then we added primers to our PCR tubes, and we added a bead. The little beads contained the DNTPs, the A's and C's and T's and G's. It contained the right buffer needed for the DNA polymerase, as well as some magnesium that Taq polymerase really, really likes. Then we processed these samples in our PCR machine, thermocycler, for 35, I said 30 earlier, it was 35 cycles at the following temperatures. Then we kept our samples at 4 degrees overnight until we came in to run the gel. Running the agarose gel was done at about 3 hours after the PCR reaction. Running an agarose gel is done to separate and visualize the DNA from the PCR reactions. In short, our samples are loaded into wells near the negative cathode. When we apply the power source, the negative current then repels the DNA, and the DNA will move through the gel down towards the positive anode. DNA is negatively charged, and so it does repel from the cathode and move towards the anode. Now, this movement is dependent on size. Small particles of DNA, small pieces of DNA move really, really quickly and will end up further down the gel over time. shorter molecules near the bottom. Larger and longer molecules take longer to snake through and move through the DNA gel, the agarose gel, and so they remain up near the top as they take longer. And so what you get is basically a size separation of the DNA fragments after you run the gel. All right. We already did all this for you, so I guess we don't need to talk about the procedure and exactly how you open the cartridge. We were careful not to pierce the gel when we loaded our samples onto the gel. This is sort of how our gel ended up looking. We have a DNA ladder in well 1. This is a collection of DNA fragments at known sizes so that we can sort of compare our unknowns. We did 3 wild type reactions. We left several lanes empty. We did 3 transformed reactions, and then again we repeated the DNA ladder. Now remember, in wild type tubes, wild type reactions, lanes that had the wild type samples, do we expect to see a band or not? Do they have the transferase gene? Will there be replication? What about the transformed? They do have the transferase gene. There should be replication. So what should we see here on this? Should we see a band? We did use a light source to illuminate the DNA fragments. This gel that we used has gold particles in it, and it will stick to the DNA as the DNA passes through, so we didn't have to stain or add any dyes. You all will find a picture posted on your Canvas site. Oh, goodness gracious. That you can download to insert into your oral communication PowerPoint. Use the ladders to estimate the band size. Note, our ladders did not turn out very well. There is something wrong with our ladder sample. There should, though, be 3 very bright bands. One at the top is 1500, one in the middle is 1000, and the lowest bright band is 500. Let me show you. Let's go back to your online resources. There will be a link that says DNA gel. Here is our DNA gel. Remember, lanes 2, 3, and 4 had wild type. There is something weird there, some sort of non-specific, not very bright band though. But the wild type, for the most part, is empty because there was no amplification and no PCR results. It didn't work. The gene wasn't there. Here is the transformed 9, 10, and 11. You see a very bright band resulting in because of the fact that the transferase gene was there. Amplification did occur, and now we visualize that DNA that was amplified here as these bright bands on the gel. You can see that here is the bright band, the 1500, here is the 1000, and here is the 500. So our amplified transferase gene falls between 500 and 1000. I would estimate it somewhere around 750, 800 base pairs or so. All right. Now, oral communications. You all should be working on an oral communication PowerPoint that includes the following components. You've got 4 weeks of antibiotic resistant data. You've got Kirby-Bauer results. So actually, let's start at the beginning. You should start out with a title. You should provide general background on antibiotic resistance, probably citing the lab manual and the Berry and Chung. You should provide some specific background on aminoglycosides and the transferase and which antibiotics you were assigned. Then you will have experimental design for Kirby-Bauer. You will have your data collection for Kirby-Bauer. You will have your Kirby-Bauer results. You will then have background for your next experiment, your MIC. You have another experiment with a new hypothesis, data collection, MIC results. Background for the next experiment, which is PCR and electrophoresis. Then you will include experimental design for PCR and gel. You will include your electrophoresis results. Then you will have combined all 3 experimental discussion, combined all 3 experiment implications, future directions, and then citations for all. All right. Now, what should you have done? You should have posted your PCR and gel electrophoresis question set to Canvas. You should have just watched this video. You should now get with your group to collect and analyze the MIC data for the antibiotics you were assigned. You should find a paper. You should find a paper on your antibiotics if you haven't already done so. You should then get with your group to determine your hypotheses and predictions for the PCR and gel experiment. And then you and your group should then analyze the gel picture. You and your group should work on the PowerPoint for the oral communication, making sure to turn it into Canvas by the end of class on March 31st. Finally, let me include one more note here. Your GTAs will be available either by Zoom or email during your regular class time. This is what this way you can actually connect with them to ask questions over the data analysis and their specific sort of requirements for you.